internacionais. E hoje nós temos a honra e a felicidade de contarmos com um professor da Johns Hopkins, uma universidade bastante renomada. E porque ele não fala o nosso idioma português, nós vamos utilizar o idioma inglês como idioma deste webinar. So thank you very much, everyone, for being here attending this webinar today. So it's a great pleasure to introduce Professor Richard Lee from the Johns Hopkins University. He's going to talk about the role of the HPA axis and the neuroendocrine system in psychiatric disorders. But today, I would like to thank you, but thank you also for give thanks for Professor Miriam Hayashi. Professor Miriam Hayashi, she's in the charge. She's in the charge of this webinar. She's part of the capstone program of UniFesp in the theme inflammation. And all of this will happen today because she was contacting Professor Richard Lee, and she was organizing everything behind the scenes. So on behalf of the Postgraduate Studies and Research Office, I would like to thank you, Professor Miriam Hayashi, for being such a wonderful researcher, such a wonderful person, and someone that contributes a lot for the development of our university. And Professor Miriam Hayashi, she was born and educated in Brazil, and she's currently Associated Professor of Pharmacology at the Federal University of São Paulo and head of the Molecular Pharmacology Laboratory at the Department of Pharmacology at the Escola Paulista de Medicina. And Professor Hayashi is also Vice Chief of the discipline in cellular pharmacology and member of the scientific board of the Intellectual Property Office at UniFesp. Also, she completed her pharmacy and biochemistry degree at the State University of Londrina in Paraná, and she received her PhD at UniFesp and postdoctoral fellowship in molecular biology at Butantan Institute, which is a Brazilian institution mostly renowned by the scientific research with venomous animal and production of vaccines. Also, Professor Hayashi is extensively involved in medical and biomedical education and is also active in multiple research endeavors, which primarily aim to characterize the association between oligopeptides and oligopeptidides and mental disorders, notably with focus on its influence in cognitive function and general symptoms. Her work broadly aims to characterize the underlying causes of mental disorders, notably schizophrenia and bipolar disorder, pursuing the neurodevelopmental hypothesis and the importance of inflammation to explain the dual influence of genetic background and environment for the susceptibility to mental disorders. This body of work has provided up to now a platform for identifying novel molecular biomarkers for diagnosis and potential new targets to treat and prevent mental disorders. Dr. Professor Miriam Hayashi has published more than 90 scientific articles on mental disorders and natural compounds for drug discovery field, besides several chapters of books and patent applications field. Dr. Hayashi was recently awarded with the prize for Fleury Group Innovation Award, thanks to the development of a method of identifying biomarkers for severe mental illness by nuclear magnetic resonance and chemometrics, protected by the patent field and regulated here in UniFesp. So, Professor Miriam Hayashi, it's a pleasure to have you here. And Professor Richard Lee, thank you so much again for taking your time to be here with us. And Professor Miriam Hayashi will introduce to you and will give all the instructions for the attendants to optimize your time and your moment here. So thank you so much. And UniFesp welcome you and says thank you for being here today with us.
Thank you, Camilo, for the long and great introduction. So I would like to introduce now Dr. Richard Lee. He's actually assistant professor in the Department of Psychiatry at Johns Hopkins University. He obtained a BS in biomedical engineering at Homewood campus in Johns Hopkins University. He obtained PhD in molecular biology and genetics, and his postdoctoral training was in the Johns Hopkins University School of Medicine. For graduate school, Dr. Lee was trained under the epigeneticist Dr. Andrew Feinberg, where his research focused on uncovering epigenetic mechanisms that underlie genomic imprinting in childhood developmental disorders. Dr. Lee then received his postdoctoral training under Dr. James Potash, currently the chairman of Psychiatry at Hopkins, and he has been applying tools and methods of epigenetics to elucidate the mechanism by which stress exposure can affect genes and pathways involved in psychiatric disorders. He also worked with collaborators to understand epigenetic mechanisms associated with drugs of abuse, psychotropic medications, and motivation behavior. And I hope that we can also straighten our connection and start some successful scientific collaboration. So I would like to thank you very much, Richard, for your for accepting my invitation, and also for the audience. And before you start, I just would like to ask the audience to make the questions by the chat, and I'm supposed to translate those that cannot express in English. So you can write in Portuguese if necessary, and then I will try to translate Richard. So we are planning to have 1 hour of presentation by Dr. Richard, and then we'll have 30 minutes for questions and discussions. Okay? So thank you very much, Richard, and the screen is yours.
Okay, thank you so much for the invitation. I'm thankful for the UNOFESP institution and Dr. Hayashi for the generous invitation. As mentioned, today I will talk about the role of the HPA axis and the neuroendocrine system in psychiatric disorders. I guess I will talk to you briefly about what stress is. It's a lot of different things, but mainly I will be talking to you about psychosocial stress and how it contributes to psychiatric disorders, and to fully understand how stress can lead to psychiatric disorders, we need a brief discussion on a field called epigenetics. So I will talk to you for a little bit on epigenetics and some of the different flavors of epigenetics, different mechanisms, and some of the tools that we use in epigenetics. And I will then switch gears to talk to you about how epigenetics pertains to stress and psychiatric disorders. And the second half of my presentation, I will talk to you about some of the case studies that were mostly done by the group here, as well as by other groups looking at epigenetics in human behavior and animal models as it pertains to psychiatry. And I'll have some afterthoughts on how HPA axis is involved in some of the substance abuse disorders such as alcoholism and drug abuse. So let's get started. What is stress? Stress has been originally defined by Hans Selye in 1936, and it's defined as a non-specific response of the body to any demand for change. And it's a very general statement, but over 80 or so years, we've learned a great deal about the physiological response to stress and what stress is made of. Now, stress for the most part is made up of, or the stress response is made up of two components. One is the autonomic nervous system, the sympathetic and the parasympathetic systems that initiate or precipitate the stress response, and then kind of returns the body back to normal. But of greater interest to me is the HPA axis. So this has a much longer impact on physiology than the autonomic nervous system. So the HPA axis consists of the hypothalamus, the pituitary, and the adrenal glands that through neuroendocrine communication mounts the stress response. And the endpoint of the HPA axis being activated is the release of cortisol, or the stress hormone. And I will also refer to cortisol as a glucocorticoid, which is the kind of a general term for these stress hormones. And so starting from the brain, the hypothalamus here in the red circle, you have the perception of stress and release of the corticotropin releasing hormone from the hypothalamus to the pituitary below. And from the pituitary through intracellular signaling, as well as physiological response, there's the release of adrenocorticotropic hormone, which when it reaches the adrenal glands, so these are the glands on top of the kidneys, release cortisol. So let me talk to you a little bit about psychosocial stress as an environmental factor. So I think I had to think about this a little bit because stress is very different from some of the other environmental factors. You know, when we think of, diet or exposure to lead or other toxicants, and even ionizing radiation, from nuclear power plants, or from sources of radiation from outer space. So in these cases, in these instances of environmental factors, you're actually physically or chemically exposed to these external factors. But psychosocial stress is a little bit different because it is not something that's directly physically affecting you. It is your perception of stress that initiates this physiological response, and it can be very subjective. So there's a stress test called a Trier social stress test where participants go in front of a large group of people, and either they do some sort of a public speaking engagement or solve I guess difficult mathematical problems. And so when you're in front of a group of people and you're tasked with these problems, it could be very stressful. So then your autonomic system kicks in, your sympathetic system kicks in, and then your HPA axis gets involved, and there's release of copious amounts of cortisol. And I say it's subjective because I could easily picture a math teacher, like a calculus teacher, going in front of the same group of people, which he or she does every day during school, and solve relatively difficult problems with ease. So then she or he would not have such a strong stress response. So it can be very subjective. And of course, the stress response is triggered even if no actual harm has occurred. And this is very different from, the stress response that we normally encountered, through evolution, life of an animal being chased by a predator. It's either life or death, right? And so there is actual potential for harm. But in this day and age, a lot of our stressors are psychosocial in nature. But nevertheless, it is an environmental factor because it is through our interaction with the environment that the stress response is precipitated. So this is the acute stress response system that involves the autonomic nervous system. So, when you're first initiating the, what's called a fight or flight response, your heart rate goes up, your blood pressure goes up, there's heavy breathing, and this is to mobilize your energy sources to tackle the stressor. Whether it's evading a prey or running away from a robber or avoiding a car accident. But these are all physiological responses that are very short in duration and for the purpose of evading an obstacle. So I am indebted to the work by Bruce McEwen, who's I consider, I guess, the father of modern day stress, as well as work from Robert Sapolsky, who's spent a lifetime working and understanding the stress response. The problem with stress is that in this day and age, stress becomes very chronic. And so we have what's called allostatic load. So this is the cumulative wear and tear on the body through repeated exposure to stress and repeated activation of the HPA axis. And so some of the mechanisms that existed to mount a quick acute stress response is now turned on chronically. And so you have, cardiovascular disease, hypertension, susceptibility to cancer. And of course, one of the things that the cortisol does is to suppress your immune system. So you have a susceptibility to infection. You have obesity and diabetes and so forth. And of great interest to me, of course, working in the Department of Psychiatry are psychiatric disorders that are often associated as consequences of chronic stress exposure. So there are sources of these psychosocial stress. Like as I mentioned, we work in the post-industrial age, the work environment with the social hierarchy, the boss that constantly is demanding more work from you, the morning commute of getting stuck in traffic or having to meet deadlines. There's relationships that we're in, whether family or, for spouses. There's financial, economic hardship. I think a lot of us have entered that kind of extra economic hardship because of the current status that we're in. For kids, there's bullying at school. And then we have the pandemic, and there are lots of people that are socially isolated as a result. And that is surprisingly, that is a potent stressor, which I will talk about later. And then, of course, in the past 4 years, we had the elected leadership. I added the phrase, make America boring again, because I think that government should be very boring, that you shouldn't have to think about them, think about the way the government works. But, unfortunately, in the past several years, there's always something going on that made headline news. So it was very stressful for many of the Americans, at least. And changing gears real quick, let me explain to you how stress hormone works. So as I've shown you before, the stress hormone itself, when it's released into the body, is based on a modification from cholesterol. And cholesterol being hydrophobic, it readily goes right through your cell membrane. And what happens is when it goes through the cells, it initiates a series of molecular reactions. And so, I'm going to get quite technical here, but the cortisol binds to a receptor called a glucocorticoid receptor, and it's usually tethered in a complex of proteins. And upon binding, it separates from that complex, it dimerizes, and then it has some additional work signaling function in the cytoplasm of the cells. But also, one of the main things that it does is it goes into the nucleus of the cell, where it binds to specific sequences of DNA called the glucocorticoid response element, or the GRE. And as a result, there are genes that are either turned on or turned off, and that initiates at the intracellular level the stress response. And one of the many genes that are made or into mRNA is a gene called or a protein called FKBP5, which I will talk extensively throughout this talk, because it's one of the few genes that I'm very interested in because of its role in psychiatric disorders. So to get into detail about how stress and stress hormone is associated with psychiatric disorders, I got into this because working in the Department of Psychiatry, I'm not a clinician. And so, it was very difficult for me to understand some of the psychiatric disorders. And for me to actually kind of understand at least some mechanism of how psychiatric disorders such as depression or bipolar disorder is precipitated, I thought, as a molecular biologist, there's stress, which is a very robust risk factor for a lot of these disorders. And dysregulation of the stress response, it turns out through, literature search, is associated with, anxiety disorder, PTSD, with depression, bipolar disorder. And one of the wonderful things about stress hormone is that you could actually measure it in the blood of, even in saliva, actually, of humans and so forth. So to be specific, what does stress hormone or glucocorticoids do in psychiatric disorders? It turns out that excess levels of cortisol and the resulting glucocorticoid resistance, so this is when your body doesn't respond well to cortisol signaling, it's often comorbid with depression. In fact, I think about 50% of the cases of depression, patients with depression have this phenomenon where they have high levels of cortisol in their plasma. And working in the Department of Psychiatry, it turns out that I learned that glucocorticoids are one of the few molecules that could actually trigger both mania and depression in bipolar disorder. And one of the diseases that I had the opportunity to work with is a rare disease called Cushing's disease. So this is where you have a tumor in the pituitary, usually, that releases, as I mentioned to you before, ACTH, then goes to the adrenal gland, and so releases copious amount of cortisol in these patients. And it turns out that about 60 to 80% of these patients have depression and anxiety disorders. And what's I guess what's insightful about this disease is that when you surgically cure them or biochemically cure their condition, by removing the tumor, the pituitary tumor, or giving them glucocorticoid receptor antagonists to block their hormone signaling, their depressive symptoms are actually resolved. So you see this nice cause and effect relationship between high levels of cortisol and the development of psychiatric symptoms. And I mentioned to you before, social isolation is a potent stressor because it turns out that we are social creatures by design, and being alone for a very long time is very stressful, not only for us obviously, but for animals that live in societies. To think about some of the relevant studies that have been done regarding the role of stress on disease, I would like to point you to the Whitehall studies done by Marmot et al. In the 1960s and 1980s. So these are two studies of British civil servants. So they have I don't know about now, but at least, they probably do right now too, because we have this kind of system as well. But there are different grades or levels of employment for government workers. So these two studies, one looked at about 17,000 employees in the British civil servant system for the first study, and then about 10,000 for the second study in the 1980s. And what they examined was looking at, they looked at coronary heart disease and, disease mortality, and compared that to employment grade level. So what they found was kind of astonishing, because it turned out that the employment grade level, so what level of employment or authority, or, the totem pole, they like to say here, what level your employment grade was, had the strongest predictor than any other risk factor for coronary heart disease. So those at the lower end of the servant system had higher incidence of coronary heart disease and mortality than ones in the higher up in the chain. And then the Whitehall study 2, the second study, also examined psychosocial factors, and they found an exact same relationship as well, where they looked at, work environment, social support, and financial difficulties, all having an inverse relationship with grade level. So this was a very large study, basically looking at job strain or psychosocial stress and the employment grade, and it goes to show that there are a slew of these disorders that are associated with increase in stress levels. And to be more specific, not only the stress, but looking at just the stress hormone itself, cortisol, or generically glucocorticoids. There was even a bigger study that was performed in 2012, looking at over several hundred thousand patients that were given corticosteroids, or basically cortisol, for diseases, autoimmune diseases, such as arthritis, asthma, lupus, and so forth. Now, these are non-psychiatric diseases, and so they compared basically, several hundred thousand patients who were given these medications for these specific disorders, and to others that did not receive these hormones. And what they found was a significant increase in the prevalence of depression. They call it hazard ratio, mania, panic disorder, and suicidal behavior. So this was a very powerful study, because you're looking at, in the human population, of several hundred thousand participants, a significant relationship between stress hormone exposure and psychiatric disorders. So looking at some of these studies in literature, I started looking at some of these genes or proteins that are involved in cortisol or glucocorticoid signaling. And so this was one of the early kind of revelations I had, my small revelation, just listing all of these genes that are involved in glucocorticoid signaling, and either in humans or animal models, there's a abundance of evidence for their role in psychiatric disorders. So now we're kind of getting into the mechanism of things, linking stress hormone exposure and some of these disorders that are associated with chronic exposure. So kind of the framework that I have is that you have environmental stressors, activation of the HPA axis, and then you also have iatrogenic glucocorticoid administration. So, for, asthma or arthritis, you have doctors, clinicians providing sources of cortisol to the patients. And so when you have high levels of cortisol, it turns on basically glucocorticoid signaling in the tissues and cells. And what you get are altered cortisol homeostasis. So your homeostatic levels of stress hormone becomes altered. It could be through genetics. So you have risk factors for how your body responds to cortisol signaling. And then you have epigenetics, which is what I will talk to you about. So this is kind of an interaction with the environment, in this case, exposure to stress and stress hormone. And then you also have protein modifications that occur as a result of glucocorticoid or cortisol signaling, which has not been studied as well, but it's definitely important for infection. And then through these modifications in the tissues and cells, you have multiple symptoms or disorders that are precipitated. So let me explain to you, or switch gears and talk to you a little bit about briefly about epigenetics. And I think that just to highlight some of the interesting case studies linking, stress or glucocorticoid exposure to psychiatric disorders, I will kind of speed through them. So epigenetics is the study of heritable changes in gene function that does not involve changes in DNA sequence. And so for the most part, it involves the structural modification inside the cell that kind of promotes or determines whether a gene is turned on or turned off. And so I'll talk to you briefly about the three main flavors of epigenetics. I think there's probably another one that's emerging soon or has been emerging. One is DNA methylation. These are modifications on specific sequences of DNA. And then there's histone modifications. So these are proteins that the DNA is wrapped around, and they're modified to change accessibility to specific pieces of DNA. And then there are non-coding RNAs. So these are your microRNAs and long non-coding RNAs that play a role in regulating gene function. And as I mentioned to you before, none of these involve changes in underlying genetic sequence or mutations. And so this is DNA methylation. It's a methyl group that gets attached, for the most part, to cytosine. So we call it CpG because it's in the context of cytosine and a guanine, where a methyl group gets attached to the fifth carbon of cytosine. And basically, a general way to think of DNA methylation is that when, for instance, in a cancer, in a tumor suppressor gene, the promoter region is usually unmethylated to check cell growth. So, these kind of white lollipops here represent unmethylated CGs. And then through, a poor diet or exposure to some mutagen, you have methylation of this promoter region of a tumor suppressor gene indicated by these black lollipops. And so then what happens is the gene is not turned on, and when you have suppression of a tumor suppressor gene, repression of a tumor suppressor gene, the cell growth is unchecked, and you get carcinogenesis. There are other flavors of DNA methylation. In the past, I think about 5 or 6 years, DNA hydroxymethylation is another relevant modification. It's very prevalent actually in the brain. So there has been an increasing number of studies looking at hydroxymethylation in psychiatric disorders and behavior. Histone modifications. About 20 years ago, there's a very influential review paper calling all the modifications the histone code, where basically some of the lysines and other amino acids become, covalently modified by different enzymes. So you could see a whole slew of these enzymes that modify histones. So histones, as I mentioned to you before, are these kind of drum-like protein complexes where the DNA is wrapped around, and it's compacting the DNA, approximately 2 meters of DNA into every single cell. So it's a way of condensing a lot of this genetic material that we call DNA into a very small space. And how that is important, obviously, in epigenetics is that these modifications are recognized by specific enzymes that basically ultimately change the structure of the way the DNA is wrapped around. So then there's so they have accessibility to transcription factors or repressors and so on that could turn on and turn off specific genes. And then you have microRNAs or long non-coding RNAs that I mentioned to you before. They work at the sometimes at the DNA level, but also at for microRNAs, they work at the RNA level to silence translation. Basically, they interfere with messenger RNAs from becoming proteins by binding to the tail end of some of these mRNAs. And then long non-coding RNAs also play a role in X inactivation. So there's plenty of epigenetic mechanisms aside from environmental factors that play a role in normal physiology. So we have X inactivation in females. One of the X chromosome is inactivated through epigenetic mechanisms. There's cell differentiation, with stem cells differentiating into, kidney cells or liver cells. So they need to undergo lots of reprogramming, not necessarily, or no changes in DNA sequence, but they need to change their programming to from being stem cells to cells that are specialized to a specific tissue. So then epigenetic mechanisms are involved, and a branching off of cell differentiation is cancer is when you have abnormal differentiation or poor differentiation of cells, and of course reprogramming of their replication potential. That involves epigenetic mechanisms as well. There's genomic imprinting, depending on whether a piece of DNA is from your father or the mother. They're turned on or off. So there's genomic imprinting disorders where epigenetics goes awry, and you have silencing of both genes or turning on of both parental genes, and then you have disease. And then, of course, in terms of interacting with the environment, there's exposure to environmental toxicants. Lead is supposed to be a very potent toxicant for neurodevelopment, even in low doses. So, lead apparently, so I'm kind of learning about some of these environmental toxicants as well. They interfere with some of the epigenetic machinery, like DNA methyltransferases, to change methylation patterns across the genome. And when that happens in the brain, you get neurodevelopmental issues. And then there are, psychotropic medications, and of course, stress. And so, epigenetic tools, I will briefly mention to you, are, the tools that have been developed, in the past several decades to understand or detect and identify some of these epigenetic mechanisms. So there's DNA methylation detection, histone modifications, and then there's, ways of looking at specific regions for epigenetic changes, let's say from stress. And then there are methods now with the deep sequencing or next generation sequencing to be able to sequence across the genome to detect epigenetic modifications. And then one of the very important tools of epigenetics, of course, is that we need to have a method of isolating very specific cell types. This is important because unlike genetics, where you could get your genomic DNA from from your saliva or from your blood or from your skin, for the most part, the genetic sequence is identical. But epigenetic patterns, of course, depending on whether the cell came from the brain, even in the brain from neurons or the supporting glial cells, or from the liver or the kidney, all of the DNA sequence is identical for the most part, but epigenetic patterns are very different depending on their cellular programming. So we use a platform called fluorescence activated cell sorting. So it's a fancy way of labeling specific cell types using antibodies and fluorescent molecules to separate out different types of cells, and then you could use those cells for epigenetic analysis. And so for detecting DNA, there's quite a few methods, such as using enzymes and so forth, but we use bisulfite conversion. So you have basically cytosines that, in the absence of methylation, turn to thymines. And then if it's methylated, it stays as a cytosine. So you can see here, when it's methylated, it stays as a C. And then when it's unmethylated, it's converted to a T. And you could see here also that some of the other C's that are not in the context of a CG are also changed. So it's a, excuse me, since you already know the underlying sequence, when you look at the bisulfite converted DNA, so this is from using sodium bisulfite. When you bisulfite treat and convert the DNA and you sequence this piece of DNA, you could tell whether previously it was methylated or unmethylated. And then I'll skip this part, but there are very quantitative methods of assessing whether a specific CG in a piece of DNA is methylated or unmethylated, but when you have a pool of DNA, there's a method called pyrosequencing that gives you a very, I guess, highly quantitative method of detecting methylation. And this is pyrosequencing. It's a quantitative machine. And then there are histone modifications where you cross-link DNA and you pull down DNA associated with these histones, and then you have antibodies against histones, specific histone modifications, and then you could sequence the DNA that came down with it to see whether a piece of DNA is modified or associated with a specifically modified histone, a piece of histone protein. This is important because then you could get across the, if you do this across the genome called ChIP-seq, you could actually get a sense of which regions of the DNA are, associated with compacted histones and other ones are open for transcription. So this is the next generation platform where instead of looking at a specific region, you could just fragment your entire genomic DNA, as for DNA methylation or with histone modifications, and then through different isolation methods. In this case, you label specific regions with a RNA probe that's attached to magnetic beads. You could actually pull out specific areas across the genome and then make what are called sequencing libraries to sequence them. So this is a particular methylation sequencing that we've done for the mouse genome. And what you get at the end are kind of these list of genes. So in this particular study here, we looked at DNA methylation changes that occur in mice that were treated with stress hormone, and then other group was treated with vehicle. And so then we basically isolate genomic DNA from both of these groups, make these what are called sequencing libraries, and then after sequencing these libraries, we separate them out into two groups and ask the question, what are the areas with the biggest DNA methylation differences? And so at the end, you have this kind of list of regions and the names of genes that are that show the biggest methylation difference between the treated and the cortisol treated and vehicle treated animals. And then you get a nice picture of some of these regions and what they look like. So these gray bars are the CGs, and then these blue dots are the vehicle treated animals, and then the red dots are the cortisol treated animals. And you can see a fairly significant difference in methylation at some of these regions. And then unlike some of the studies where you look at one or two genes, if you look at many genes across the genome, in this case we looked at probably over 10,000 genes or regions of genes across the genome. You could then identify what these genes are and what pathways they might be involved in. So then you could look in, you could do like what's called a pathway analysis in blood and in the brain, and you could see in both of these cases the similar types of pathways that are implicated from cortisol treatment. As I mentioned to you before, there are some obstacles in psychiatry when it comes to doing epigenetic analysis. And so this plays right into kind of the need for biomarkers. And so, in introduction to Dr. Hayashi, she's involved in, looking at biomarkers of, schizophrenia. And so, I think epigenetics also, we are in need of biomarkers when it comes to psychiatric disorders. And in doing so, we have several obstacles. One is, tissue heterogeneity. Of course, we're interested in the brain, but as I mentioned to you before, depending on whether it's brain or the supporting glial cells, there are specific and different epigenetic signatures. And then, of course, why do we need biomarkers? It's because some of the tissues that we are interested in, where the disease is occurring, is not accessible. I mean, I've done some work with postmortem brain tissues, but in these cases, the patients are already dead, and you're trying to identify some sort of disease relevant signatures in these brain tissues. But to really help the patient population, you need biomarkers of things that are occurring in the brain while the patient is alive, right? So then you need biomarkers, peripheral biomarkers, because the brain is inaccessible for the most part. And some of the obstacles that we have with biomarkers, and of course with epigenetics in psychiatry, is that there are lots of confounding factors. So we have age, sex, and medication, comorbidities. All of these affect epigenetic signatures. Well, not only when you're looking at it just for epigenetic modifications, but of course as biomarker potentials. We know that for those of you who are familiar with epigenetics, we know that there's a large epigenetic change that occur as you age, and there are differences in epigenetics between males and females. And then of course medications cause epigenetics as well, epigenetic changes as well. And this is what I mentioned to you before about obtaining homogeneous population of cells. In particular, you can see here, this is from a brain tissue of Alzheimer's disease patients. And for the sake of demonstration, I went through and looked at a specific gene that's specific for the glia. And then I separated out basically DNA from neurons versus the glial cells. And you can see here, the y-axis is percent methylation. And then these are, on the x-axis, the different positions of CGs. So there are 7 CGs that are consecutive. I'm looking at methylation by power sequencing of, in blue, neuronal cells, and then with glial cells. And you can see a world of difference in epigenetic modification, and it's coming from the same block of tissue where you cut it out from the postmortem brain, and then you process it further to identify these or to isolate these 2 species of cell types. So other than, I guess, in addition to stress, there are lots of evidence for the role of epigenetics in psychiatry. We have, monozygotic twins. Some of them are discordant for psychiatric disorders, and then we have onset of illness in adolescence for some of these disorders like depression and bipolar disorder and schizophrenia. You don't see cases of these disorders as much in early childhood compared to adolescence and young adulthood and so forth, right? And there's also the episodic nature of illnesses like bipolar disorders. They have they cycle through depression and mania, and so then there are presumed to be epigenetic factors that are involved. And then of course there's the relationship to environmental factors. I'll present to you some of the case studies briefly about some of these instances, and then even mechanism of action of medications. Now we have ketamine and psilocybin, which are thought to be or shown to be rapid acting antidepressants, but some of the other medications take a long time to achieve their therapeutic effect, and it has been thought that it takes a while for some of these medications to actually cause long-lasting changes, possibly through epigenetic modifications. So these are some of the examples of where epigenetics is involved in the stress response. One of probably the most important things I found working with animal models of stress and even cell lines is that exposure to stress hormone can directly change DNA methylation. So other people have looked at histone modifications, but I've focused mainly on DNA methylation because the changes in DNA methylation to stress hormone is very profound, as I will show in the next slides. So I've focused on a gene called FKBP5. If you remember, oh, there it is. If you remember from the next slide that I've shown you previously, it is one of the genes that are, or proteins that are involved in modulating glucocorticoid signaling. And it turns out that, ever since one of the first studies published by Elizabeth Binder and her group at Max Planck Institute, genetics and epigenetics of FKBP5. It's a gene that's been implicated in a whole slew of psychiatric disorders, through mood disorders, anxiety disorders, and even addiction as well. And here you can see why FKBP5 is so important, because after you have glucocorticoid signaling and activation of glucocorticoid response genes, one of the genes that are transcribed is FKBP5. And so what happens is when you have more of this gene transcribed and translated, it acts as a intracellular negative feedback on this entire glucocorticoid signaling. So I mentioned to you before that in about 50% of the cases of depression, is associated with hypercortisolemia and glucocorticoid resistance. So this is one of the mechanisms of how glucocorticoid resistance is achieved in depression. It's because you have high levels of FKBP5 being made from, I guess, repeated stress exposure in these cases of depression. And what it does is it blocks additional glucocorticoid or cortisol signaling. And what happens is you start accumulating lots of stress hormone in the plasma. And then because of this signaling, the body is no longer as sensitive to glucocorticoids or cortisol as before. And to be specific, some of these genetic variants of FKBP5 are associated with depression, either recurrence or response to antidepressant treatment. There are SNPs that are involved, actually the same SNPs or genetic variations are involved in bipolar disorder. There's also gene environment interaction with exposure to childhood adversity on a risk for PTSD. And so in the past, probably about 10 years since the genetic variants in FKBP5 have been implicated in psychiatric disorders, it turns out that there's been a whole slew of studies, probably several hundred now, that have implicated FKBP5 in these various psychiatric disorders. And I think it's a testament to the role that stress and the neuroendocrine system plays in the precipitation of psychiatric symptoms. And so this is a small part of my work looking at epigenetic patterns. So this was, it's a very simple experiment done in a cell line. This was obviously replicated in an animal model as well, but you could see with treatment with stress hormone, in this case CORT, or corticosterone for the rodent version of the stress hormone cortisol, you can see here that animals that are treated for about 4 weeks show, I guess, different increases in the expression levels of this gene. And then when you take away the hormone during the recovery period, expression goes down to normal. But then when you have when you're looking at DNA methylation patterns, so this is percent DNA methylation in the y-axis, and during the treatment with stress hormones, so this is in a cell line treated for 7 days with stress hormone. We've also done the work with mice for 4 weeks. You can see here a gradual decrease in methylation, and even when you take away the stress hormone from the media, you can see that the methylation persists. And so, and then in figure C is a negative control region that is not responsive to stress hormone, meaning that it doesn't have a binding site for the glucocorticoid receptor. So what this tells me, and also what the animal work that I've done that complements this, what this tells me is that, the expression levels may go back to baseline after treatment with stress hormone, but the epigenetic modifications persist. And how is that important in psychiatric disorders, especially when it comes to stress, is that in the bottom panel here, is that if you have normal methylation and you are treating the cells with stress hormone, there's an increase in the expression level of this gene. But when you've had previous exposure so that your methylation levels are already decreased, second exposure to a stressor has a even more robust response to this gene. So the first exposure, I mean, in this case, it's a cell line, but you could actually think of it in terms of a stressful exposure during, let's say, childhood. And in the brain, especially when it's, undergoing maturation and development, you could see that there's long-lasting, persistent epigenetic damage being done, so that later on, when you're exposed to another stressor, your cellular response, in this case, affecting glucocorticoid signaling, actually is more robust. And this is a little bit complicated, so I'll just kind of go briefly summarize. What this shows is that the methylation change. So this was in a article that was published actually last month in Epigenetics, by my group. What this shows is that the ability to undergo stress-caused epigenetic change is dependent on replication. So this is kind of consistent with what has been known as the Barker hypothesis. So this is when this was basically stating that conditions in utero, when you're in the womb, the amount of nutrients and calories that are available for your development in the womb actually has a impact on metabolic disorders later on in life. But in this case, this might actually explain in the context of psychiatric disorders, the importance of the prenatal environment and for consequences later on in life during adulthood. So, in a nutshell, in summary, what happens when you lose DNA methylation with stress hormone exposure in the FKBP5 gene is that in panel A, you have acute exposure, so you have glucocorticoid receptor binding to this region. And you can see here there's varied methylation status across this area bound by some of these transcription factors. And then, that turns the gene on, in this case, FKBP5, but the strength of the transcription is rather weak. On the other hand, if you've had repeated exposure or you had chronic exposure to stress, either early on or even as an adult, you could see here that the methylation levels are decreased, or the areas become demethylated. So you have loss of some of these factors. In this case, this is a repressor called MECP2 that's involved in Rett syndrome. But these very important neurodevelopmental proteins recognize methylated CPGs or CGs, and then when it's unmethylated due to stress hormone exposure, it's no longer bound. And so then your transcription is actually of FKBP5 is actually much more robust. So thinking about the role that FKBP5 plays in psychiatric disorders and the need for possible biomarker development, we've started to look at the role of some of these epigenetic patterns as potential biomarkers. So one of the things that we did was to look to see whether epigenetic patterns that we detect in the blood could actually predict some of the processes in the brain. But first, we wanted to see if it correlates to any of the other symptoms of glucocorticoid or cortisol exposure. And so what we did with animal models is that we wanted to look to see whether methylation levels actually correlated with a month worth of stress hormone exposure in the animals. And so we did several correlation analysis where we looked at, the plasma cortisol level. So this is each dot represents an animal, and it's a summation of all the different times that we took hormone measurements from their blood and compared that to DNA methylation patterns at the end of that treatment period. And so you can see a fairly strong correlation between stress hormone levels during that period and the methylation level at the end. So this is for 2 CpGs, and then we also looked at for FKBP5 expression, and then we looked at some of the other parameters that are affected by chronic stress hormone exposure, such as organ weights of the thymus, of the adrenal glands. We looked at spleen weight and visceral fat. So remember, from chronic stress hormone exposure, there's changes that occur at the periphery, such as obesity and diabetes and so forth. So we were confident that the DNA methylation pattern that we're detecting could actually reflect some of the other physiological changes occurring in these animals. And then this was actually presented when I was at Sao Paulo several years ago. I think it was 2014. Looking at the correlation between DNA methylation and some of the processes that are occurring in the brain. So in this case, we're looking at DNA methylation of the FKBP5 area against gene expression in the hippocampus, as well as DNA methylation in the hippocampus. And so there was a fairly decent and significant correlation between what we could get from the blood and what's occurring in the brain. So then, I like to present to you some of the case studies that we've been working on, looking at stress hormone response in the human samples as well as some animal models. But one of the things that we did was then, if we could successfully detect methylation in the blood to predict 30 days of stress hormone exposure in these animals, can we also do that for humans? So we recruited about 100 or so healthy humans. This was a very, I guess, a long endeavor, and to see whether methylation levels in the blood could actually predict about a month of cortisol measurements in about 100 participants. And we measured this by getting their saliva cortisol levels morning and night consecutively for 30 days. So this was not monumental, but at least for me, it was a very difficult study in collaboration with Gary Wand, who's the endocrinologist who kind of oversaw this whole project. And what we found was that, it's not as strong, but there's a fairly decent correlation between amount of stress hormone exposure, or stress hormone that we measured in these participants, and the methylation levels. And then we using some of the deep sequencing or next generation methods that I mentioned to you previously, we then looked at some of regions across the genome. So this is using a platform called Agilent SureSelect MethylSeq platform. And we looked at about 33.7 million CGs across the genome of specific regulatory regions. And, we identified methylation patterns that were associated with 30 days of emotional stress, bedtime cortisol, and awakening cortisol. And we're also able to come up with some of the pathways that are involved. The pathways are not as robust in terms of analysis compared to the animal models, because as I mentioned to you before, there are lots of these confounding factors, I think, that really reduced our signal to noise ratio. But similar to what was done with the animals, you could see that there are specific regions in other areas of the genome. In this case, this is a GABA receptor, one of the subunits of GABA receptor that's involved in the neurotransmitter systems that show differential methylation between people with high versus low emotional stress. And then we use, as I mentioned to you before, a method called pyrosequencing to quantitatively and independently verify that these methylation differences were actually real. And then this was a work done by Elizabeth Binder and her group, as I mentioned to you before, on the FKBP5 gene. And in this case, we are looking at the contribution of genetics. So, as I mentioned to you before, the epigenetics plays a big role in the stress response, but there's also an aspect of genetics where your genetic variations can determine your susceptibility to stress-related psychiatric disorders. In this case, you could see a much stronger activation of the FKBP5 gene if you have the risk allele in B compared to the protective allele. So this was a case of where individuals that were exposed to childhood trauma and had also the risk allele showed a much higher activation of FKBP5 compared to those that were not exposed and those also who had the protective allele. And then this was a very small study that we did in cord blood samples. Remember, I told you about the kind of the Barker hypothesis about the importance of the prenatal period. Here we looked at several of the CPGs important, as well as the risk allele. And it turns out that if in the cord blood DNA of mothers who were exposed to or who had anxiety or depression, there was a much higher methylation level in a subset of our samples that had the risk allele compared to the protective allele. And then, as I mentioned to you before, stress plays a prominent role in bipolar disorder. This was done with Dr. Jenny Willauer from University of Iowa, looking at FKBP5 expression across the gene in control postmortem brain compared to postmortem bipolar samples. And you can see here there are expression differences as well as methylation differences. And there's a fairly decent correlation between methylation and expression, suggesting a potential role for this stress response gene in bipolar disorder. This is another fancy, I guess, for me, animal study that I did looking at directly actually modifying DNA methylation of the FKBP5 gene using the gene editing tool CRISPR. So, that's a lot of people are using these tools, these very powerful tools now to edit the genome, but they also have ones where instead of the nuclease, they have the enzyme that modifies DNA methylation or histone modifications attached so that you could actually site-specifically modify the epigenetics of a specific region. In this case, it's the basolateral amygdala that was targeted for FKBP5 methylation. So we've actually introduced the Cas9 DNA methyltransferase construct into these brain regions to site-specifically modify a region within the FKBP5 gene. And what we found was that after modifications, these animals actually had lower levels of FKBP5 expression, and they behaved differently than the negative control animals in a test of anxiety-like behavior on the elevated plus maze. And finally, this is a fairly recent endeavor of mine, which is to look at the role of HPA axis in another psychiatric disorder, which is substance abuse. I was rather surprised to find that the HPA axis plays a prominent role in some of these substance abuse disorders like alcoholism and opioid use. And it turns out that these disorders are very comorbid with psychiatric disorders. It goes both ways. So if you look at people who have addiction, they often have psychiatric disorders and vice versa. And it turns out that it's because, in part, these disorders share same set of genes that it turns out that they're activated by the glucocorticoid signaling or HPA axis activation. And one of the important things that I found through looking at the literature for human studies and animal work is that when you actually get rid of their ability to mount a stress response, it significantly alters their craving for drugs. So this is something that I'm trying to get to the bottom of probably in the next couple of years, to try to see what kind of role stress plays in some of these substance use disorders. And so I'm going to leave you with kind of a summation of what I think happens when you are chronically exposed to stress or stress hormone, and how psychiatric disorders develop. So you have exposure to stress hormone and glucocorticoids, and as in the case with the FKBP5 gene, which regulates intracellular glucocorticoid signaling, you start getting alterations epigenetically with chronic exposure, right? And then that affects, by affecting the systemic stress hormone levels, it's affecting a group of target genes that are important for disease. And so it's a kind of a cyclical vicious cycle where these HPA axis genes, some of which alter glucocorticoid signaling, such as FKBP5, and another gene that I study is the glucocorticoid receptor. And by altering them, they're continuing to alter the cortisol dynamics. And then when it's altered at the systemic level, it's affecting specific tissue-specific processes, especially in this case, the brain. And this is a kind of a rather complex figure which outlines the interconnectedness of the neurotransmitter systems. So one of the things that, another thing that I've been working on is to see which of these HPA axis regulated genes are involved in neurotransmitter function. It turns out there's quite a few. There's, dopamine signaling, like tyrosine hydroxylase. There's the serotonin transporter, serotonin receptor. Many of these genes that encode neurotransmitter transport synthesis and receptor signaling are regulated by glucocorticoid or stress hormone signaling. And because they are, because these systems, these neurotransmitter systems are interconnected, meaning that, in different regions, they have neurons that project to different regions of the brain carrying different neurotransmitter systems. You can see how even a dysregulation by stress of any one of these neurotransmitter systems can affect other brain regions and other neurotransmitter systems. So one of the things I did not cover in this presentation is that there are a whole slew of these genes that are also epigenetically modified by repeated or chronic stress hormone or stress exposure. So it gives you a kind of a framework of how repeated or chronic stress and alterations of the HPA axis leads to psychiatric disorders. Thank you.
Thank you, Richard. Can you hear me?
Yeah.
That was awesome presentation. very, very instructive and very interesting. And
Oh
I'm
my
pretty sure
gosh.
the audience really appreciate it.
Thank you.
And I have a couple of questions for you in the chat. And if you don't mind, I would like to start with some of them. And first of them, actually, I think it's a general answer. So it might give a chance for you to summarize whatever you have presented today.
Yeah.
But, it's asking, in the current knowledge, what is the quality of evidence for association between psychiatric disorder and epigenetics?
Yeah, so there's actually strong or quite a bit of evidence between epigenetics and psychiatric disorders. Uh, even aside from stress, there's been a whole number of studies by different groups, looking at either in the postmortem brain or peripheral tissues of psychiatric patients. You have schizophrenia, uh, bipolar disorder, depression, PTSD, and they have found a whole slew of epigenetic modifications or epigenetic differences. DNA methylation, and there's a group up in I think Mount Sinai. Akbarian is a very prominent researcher in histone modifications, looking at postmortem brain tissues of schizophrenia, and found a large amount of evidence to show dysregulation of epigenetic patterns in psychiatric disorders. Oh, you're muted.
Yeah, thank you, sorry. And I would like also to complement this question with my own question. Actually, I can understand the importance of those DNA methylation and genetic modification that could control the expression of some specific proteins. Do you believe that it is possible to have a long-term regulation by the proteins that are expressed after DNA methylation, for example, or those that are not expressed anymore? I mean, I was thinking about a long-term response by modified protein or by new status of the animal after those modifications induced by cortisol or by stress.
Yeah, so yes, you're like coming in and out, but I think I got the gist of your question. Yeah, so protein modification, and since I presented this to you last year, I've also took the opportunity to learn more about protein modifications in with stress and epigenetics, and there isn't as much, but actually found a lot of evidence for the role of protein modifications in psychiatric disorders as well. So the glucocorticoid, I presented in one of the slides earlier on. There's role of genetics, and there's epigenetics, and there's protein modification. So I specifically added that because I started finding some quite a bit of evidence showing that protein modification is another function that's associated with the glucocorticoid signaling. So not only does the glucocorticoid receptor affect gene expression and epigenetic modification, but in the cytoplasm, it also helps with other cofactors to phosphorylate or dephosphorylate specific proteins. And then these proteins could go and even in the chronic sense perform whatever function that they're doing at the cellular signaling. And alternatively, some of these genes that are transcribed, they're made into mRNA. And so this is a limitation of my research, which is because it's convenient for me to look at mRNA. That's what I measure, but you know very well, of course, because you work with proteins, that that there's not a good correlation between amount of mRNA and proteins. So mRNA could actually disappear after a little bit, but depending on the protein, some of the proteins could last much longer, right? And so in the chronic sense, I think that depending on the half-life of some of these proteins, they could have a long-lasting effect even after the mRNA has been degraded.
Yeah, actually, in this case, I was wondering about your protein FPK5. I think I just would like if this protein could be, for example, higher expression, and then they could have a long-term effect. Because those proteins are controlling or regulating the glucocorticoids receptor, right?
Yeah. So I do some protein work, usually looking at histone modifications, but I actually did look at FKBP5 protein levels after glucocorticoid treatment. Yeah, so that's one example where the protein actually stays around for a much longer period than the mRNA transcript.
Yeah. So actually, I will come back to the chat. There is another question. It's from Joel. You may know him. And he's asking that since stress exposure represents a major environmental risk factor for schizophrenia and other psychiatric disorders, and HPA axis dysregulation appears a consequence of social stress, but not all patients experience the same kind of event. They are not all have psychosis. So could these X be regulated by other factors like cytokines or LPS from the microbiota?
Yeah, of course. Is he talking about the differential response to stress?
Yes.
Okay.
Variation in response to distress.
Yeah. So one of the, I find it to be very complex and interesting. So one thing that I did mention that results in a differential stress response is genetics, right? So one of the slides that was not, I did some of the work with the cord blood, but the previous slide with, a large group of participants looking at genetic variation and their stress response definitely implicates at least genetics as a factor, right? There's a risk allele. If you have the risk allele and then you have exposure to in this case, childhood trauma, then you are more likely to develop psychopathologies later on compared to somebody who's not exposed, even with the risk allele, or somebody who's exposed but has a protective allele, right? So there's a differential stress, response later on. And talking about some of the other factors, of course, the cytokine response is another consequence of stress hormone exposure. So you have usually glucocorticoids are anti-inflammatory, right? You have hydrocortisone, another glucocorticoid that you rub on your skin when you have inflammation, when you have wet swelling, and it makes it go away. But it turns out that there's other pro-inflammatory cytokines that are upregulated by stress hormone signaling. It depends on I guess the condition of the tissue and the cell type, but IL-6 like TNF alpha in some studies are upregulated by stress hormones, whereas many of the other immune system is suppressed, right? So you could easily envision people having a differential immune response, and the role of inflammation in some of these psychiatric disorders as well, having a different factor, different response because they have different contributions. Yeah.
Actually, I would like to complement this question, asking you if you have an idea or you have ever read something about basal and long-term stress, like if the animal or even the person, the human, is under stress, it's a basal stress, if they respond differently for the cortisol for a trauma.
Are you talking about humans or animals?
In both cases, if they are going to respond in the same way for the ventilation.
Yeah. So, remember, so this is as you said, complementing the previous question. Yeah, there's definitely a differential response, right? So I could think of what is that? The social defeat stress work done by Eric Nestler, where he clearly identifies, groups of animals that are, resilient versus susceptible to stress, right? So they undergo the exact same stress regimen, and at the end, they have very different stress response in terms of their interaction with other, social avoidance is one of the tests that they use to assess resilience or susceptibility, right? And so they have a very different response. And then with humans, of course, in addition to what I mentioned to you before, when you give people the social stress test, obviously, it's not ethical. We are already highly stressed enough, but it's unethical to chronically torture somebody, like a human being. But, there are some acute stressors that you could give people. And, there's a huge range in the response that you get. And it depends on their genetic makeup. And then, as you mentioned, maybe their cytokine profile, even the microbiota, the interaction with, the gut bacteria. But, like I said, the psychosocial stress, at least for humans, I think, is also very subjective, So, yeah, doing these kind of online meetings, sometimes I start getting, panicky because, I don't have a face to look at, when I'm just reading, looking at those slides, but other people might do just fine, So it's a very different response for humans too.
I think you can close your presentation because they close it here. So actually you can have your normal screen.
I think it's closed.
Yeah, it's closed. Someone closed for you.
Okay, yep.
So it might be better. At least you can see me, but we can see you.
Yeah, that's why I feel so much better.
Okay, it's all you have for now. I'm sorry. But okay, so actually what I was asking is that you mentioned about previous contact with cortisol, for example. If you have a previous sensibilization, then you could possibly have a stronger effect later on. So I was just thinking that those that are resilient animals, for example, or even human, under long-term stress in a lower background, if they are going to respond more strongly to a new stress situation.
I'm sorry. If you're saying if you are exposed to long-term stress before,
yeah, if you're going to be more sensitive to new stress, a new trigger.
Yeah. So this is something that I've also been researching recently. So if you have had chronic stress for a long time, and I think, obviously some of the work that I've done is, basic science, right? It's in the cell line, and I'm now, trying to look at some of the behavior in the animals to show that it's actually the case that when you have a much stronger response later on, that, you're more susceptible, right? But I think that there's some also recent evidence suggesting that, if you have maybe not chronic stress, but acute stress, repeatedly, in a training fashion, then you may be resilient to stressors down the line. So, it's still a bit unknown as to, what magnitude and duration of the stressors actually precipitates a more harmful stress response and what leads to resilience. I think that's a very important topic, especially, for the military, right? Because they want to know, somebody who's been through combat, how, what factors make it more likely for them to, be gripped with fear, in the second time they go on duty, tour, versus somebody who, might not think too much of it, right? So that's an active area of research that I'm not quite aware, I'm not quite sure, but in terms of some of the molecular aspect of it, if you look at, some of the epigenetic changes that occur to change gene function, it definitely not only work by me, but from other people too, that, these previous exposure events act as kind of a primer. They prime your response for the second one to be more robust.
Yeah, exactly. Coming back to the audience again, there is another question from Rafaela. and she's thanking for the class, and she's asking if you ever found any evidence in the literature related to stress and ADHD.
In what? I'm sorry?
Between stress and ADHD.
Oh, I'm sorry. Between stress and
It's a hyper.
oh, ADHD.
Yes, ADHD. Yeah.
I think there's some evidence for that. So that's one of the weird things that we found, but we never pursued because the evidence obviously we need more replication. But the cord blood samples, right? The cord blood sample work that I briefly showed you guys, where we looked at the cord blood DNA of FKBP5, for methylation, right? It turns out that in the group that had, in these methylation patterns in a kind of a sequence-specific way, and that had mothers who had depression and anxiety. So, in pediatrics, it's called toxic stress, right? It turns out that there's I think it was like probably around 50% of some of these children that came from being exposed to this maternal toxic stress had ADHD. That was a kind of something that we weren't looking for, but we thought that was really interesting, but we never pursued it. And sure enough, I think that there's a lot of not a lot, but there's a lot of evidence for stress in utero and some of the consequences. I'm not quite aware if the evidence is really strong, because I don't know the literature between stress and ADHD, but I thought that was a weird, significant finding that we had, especially with that cohort.
Okay, thank you. We have another question. It's related with premature or early aging in psychiatric disorders. So like schizophrenia patients can dead prematurely. We know that there is some correlation with the length of the telomere, but is there any kind of DNA methylation related with early aging?
Yeah, so so actually, I've actually some of the animal models that I've used, and human samples. So there's a manuscript that I've been sitting on for like a year, looking at models of stress hormone exposure in mice and rats, and in human Cushing's, and in the healthy human cohort that I mentioned to you guys in the slides. In all of these cases, there's a very nice a significant relationship between the amount of stress or glucocorticoid exposure and telomere length. Now, because of the pandemic and because the reviewer asked for comparison between the qPCR method and the old radioactivity method of measuring telomere length, we took a long time because of the pandemic reasons. But yeah, so absolutely, I think if telomere is really a measure of premature or aging, then definitely we found across multiple species evidence for excess glucocorticoid exposure and shortening telomere length. And there's also probably even more accurate or powerful method of assessing epigenetics. So this is the epigenetic clock by Dr. Horvath at UCLA. So looking, he uses the Illumina epic array or the methylation array, looking at at that time 450 CPGs and identifying very specific CG methylation patterns associated with aging. So since the publication of his groundbreaking paper, there's been a whole slew of work using that platform, for various diseases to look at premature aging. And so I did not follow that literature as closely, because I think that the telomeres, and even work that I do with telomeres, some work that I've done with telomeres, and some of the work that's done with epigenetics is aging is kind of indicative of kind of general disease burden. So I don't think it's very specific to psychiatric disorders, but I think there's definitely lots of studies. If you look up Horvath clock and epigenetics, you'll definitely find a lot of papers looking at psychiatric disorders and non-psychiatric disorders and premature aging. There's definitely evidence for that.
Yeah, I do agree with you, because most of the disease is actually finished with oxidative stress, and this is closely related with aging. So it's very difficult to indicate this for a specific disease. But in the case of psychiatric disorder, we have also the use of antipsychotics that are quite strong medications with a lot of side effects. So they might have the effect of the drug and not only of the disease.
Right. And, some of the work recently done by me and previously by other people, I think also, now that you mention psychotropic medications, is that even many of the SSRIs, that are supposed to work at the serotonin transport level are actually affecting glucocorticoid signaling. So I'm sure that's not their intended effect, but I think there's now several studies that show that treatment with antidepressants, especially SSRIs, normalize glucocorticoid signaling in animal models of stress and in psychiatric patients.
Great, Richard. So if there is no any more question, I just would like to thank you for your great presentation and for this long discussion. You might be hungry because of the time.
Yeah.
So thank you once more, and I hope we can keep in touch.
Yep, of course.
Have a nice lunch.
Yeah, thank you so much for this invitation.
Thank you.
It was my pleasure. Thank you. Bye-bye.